Hi everyone, this is Ms. Moriarty. I'm here to discuss with us topic 8.12 as well as 8.13. Now in 8.12, this is all about lethal dosage, which we started talking about in class today. We know that lethal dose 50 or LD50 is going to be the dose of a chemical that's going to be lethal to at least 50% of the population of a particular species. We can see lethal doses often graphed like this we see in the picture here. Independent variable we see is usually the concentration of some form of chemical that can either be added via food, water, maybe air. And the dependent variable is going to be the response measured in the organism. Usually it's death or sometimes maybe even impairment. Now, LD50 data is oftentimes expressed as either mass or grams or milligrams per body unit of mass, oftentimes expressed as kilograms. Sometimes we can also see it in parts per million if we're referring to elements or compounds found in the air, or sometimes mass per volume if we're talking about in water of some sort of blood volume. Now in 8.13, this is all about a dose response curve. We can see the data from a dose response study was graphed with the percent mortality or other effect on the Y versus the dose concentration on the X. The dose response curves are usually shaped kind of like an S-shaped, very low mortality at low doses, but a very rapid increase in mortality as dosage continues to increase. We could see that it's going to most likely level off near 100% mortality at incredibly high dosages, depending upon the element compound. Now, something we didn't necessarily mention in class today, but you will investigate this tomorrow in the activity. There's a couple different names for this type of phenomena. So we know lethal dose 50, that's lethal to at least 50% of the population. We can also have what's called ED50. And that refers to the dose concentration of that toxin that causes a non-lethal effect. So there's still some sort of biological response. It's just not necessarily lethal. So that could be something like infertility, paralysis, cancer, vomiting. And that we're going to see it be experienced in at least 50% of the population. A good example of that is actually a case study. I know we didn't talk about it in class, but a classic case study is actually the concentration of atrazine, which is a popular pesticide that was once used that is still found in water because we know it's very difficult for it to break down. And it is frogs that actually are highly affected by concentrations of atrazine, and we can see 50% of frogs become infertile at specific doses of atrazine exposure. We are also going to see a very similar kind of S shape as we did with LD50. We're going to kind of see a similar thing with effective dose 50. But at lower dose concentrations, we're going to see a less of an effect. And so what does this mean for then human health? Well, dose response studies are used for toxic chemicals. We know it's not done on humans. Oftentimes it's done on other types of organisms like rats or maybe even cockroaches. But this is done on these types of organisms so that we can figure out, well, how would it affect a human in this case? Because we want to determine the maximum allowable levels for humans. And that also brings me to a couple other vocab words that we need to know and need to be familiar with is acute versus chronic. So if you were to be exposed to certain pollutants at certain periods of time, that may be acute or that may be chronic, right? Acute means for very short periods of time versus chronic, which means you are exposed for very long periods of time. So most dose response studies are considered to be acute, right? They only measure effects over very short periods of time. Usually they're very isolated. So they don't necessarily measure ecological effects of those organisms. For example, if one organism in a food web were to be highly affected by a certain concentration of a toxin and that organism begins to die off, we're going to see a kind of a trickle down effect in the food web. Now, chronic studies are much longer term and follow developmental impact. So they can actually follow what's going to be the impact of that toxin from the development in a fetus all the way up until adult. So we can see, right, the study of fish from maybe hatchlings all the way to adults, to then their sexual maturation, in where and how that toxin is going to affect them. And so quick multiple choice. So based upon the data that we see in the graph to the left, what is the best description of this relationship? So at this point, please pause the video and see if you can answer. All right, so at this point, you should have paused the video and let's see if we are correct. So here, based upon our data, the best description of the relationship that we see in the data here is as the concentration of anastrol increases, the percent induction of breast cancer is going to decrease. All right, guys, that's it for this video. Please make sure as you watch, you please take notes and please mark it as done.